Hello students and my dear friends, myself Madhu Vancha. In this session, we'll discuss some of the parasitology multiple choice questions which will be helpful in the lab technician recruitment examination. First question, most severe malarial infection is caused by option A, Plasmodium falciparum, option B, Plasmodium malariae, option C, Plasmodium ovale, and option D, Plasmodium vivax. So if you see all these options, like all these species, they causes malarial infections. But which one of the malarial parasite, which one of the species causes most severe infection? If you see, the right answer is option A, Plasmodium falciparum. So Plasmodium falciparum is most pathogenic Plasmodium species which infect the humans. So here there are no relapses in Plasmodium falciparum infection, but if you see, relapses occur in Plasmodium vivax. And not only that one, so Plasmodium falciparum, it invade all the type of erythrocytes, like all the ages of erythrocytes, whether it is young RBC or old RBC, so they invade it. But if you see Plasmodium vivax, they invade only young erythrocytes. And not only that, Plasmodium falciparum causes complications like pernicious malaria and black water fever. So here the right answer is Plasmodium falciparum. Next question, scientist who discovered transmission of malaria by Anopheles mosquito. Means who discovered the malarial parasite, means that is caused by the Anopheles mosquito. Option A, Louis Pasteur. Option B, Ronald Ross. Option C, Robert Koch. Option D, none of the above. Here, the right answer is option B, Sir Ronald Ross. Sir Ronald Ross, he demonstrated that mosquitoes of the Anopheles species. So Anopheles mosquito are the vectors of malarial parasite. So he did his research in India only, in Hyderabad only. In the year 1902, he awarded with the Nobel Prize. Next question, malarial parasite discovered by option A, Louis Pasteur, option B, Ronald Ross, option C, Leverian, option D, Robert Koch. So here, so previous one is who discovered the who told the malarial parasite is transmitted by Anopheles mosquito. Here the question is malarial parasite discovered by. So what is the right answer? Here that is option C, Leverian. So if you see Charles Leverian, in the year 1880, he discovered the malarial parasite. And Ronald Ross, he told that it is transmitted by the female Anopheles mosquito. He demonstrated, he proved that. So here the right answer is option C, Charles Leverian. Next question, serological test used to detect serum antibodies in visceral Leishmaniasis. So what is Leishmaniasis? So visceral Leishmaniasis. So Leishmania donovani, it causes visceral Leishmaniasis, or it is also called as Kala Azar. So here it is detected by option A, DAT, option B, ELISA, option C, IFAT, option D, all of the above. So what is meant by DAT? That is direct agglutination test. And option B, ELISA, enzyme linked immunosorbent assay. Option C, IFAT, indirect fluorescent antibody test. So here what is the right answer? That is option D. So all these tests, they used to detect the serum antibodies in visceral Leishmaniasis. Next question, the following Plasmodium forms are found in human blood. So which of the Plasmodium forms are found in the human blood except option A, ring form, option B, trophozoite, option C, gametocyte, option D, sporozoite. So here the right answer is option D, sporozoite. So if you see other options like a ring form, trophozoite, gametocyte, these three they appear in the blood. But if you see sporozoite, sporozoite is the infective form to the humans. And where it is found? It is found in the salivary gland of the female Anopheles mosquito. So it is present in the salivary gland of female Anopheles mosquito. It is not identified in the blood. Next question. Specimen used for diagnosis of trichomoniasis. So what is meant by trichomoniasis? Trichomoniasis, it is caused by Trichomonas vaginalis. So it causes infection in the females, as well as it causes urethritis in the males. So here it is the only parasite which causes sexually transmitted infection. So here which specimen is used? Option A, sputum. Option B, stool. Option C, vaginal discharge. Option D, none of the above. So here the right answer is option C, vaginal discharge. So as I told you, so Trichomonas vaginalis, it is the only parasite which causes STA, sexually transmitted disease. So in case of trichomoniasis, so generally we collect the vaginal or urethral discharge. Next question. Rapid and accurate method of staining to demonstrate trichomonads. Option A, iodine stain. Option B, DFA test. Option C, Leishman stain. Option D, Giardia stain. So just now we discussed what is meant by trichomonads. Trichomonads are Trichomonas vaginalis. So it causes trichomoniasis, and it causes trichomoniasis in the females and urethritis in the males. So as we discussed, in case of Trichomonas vaginalis, we collect the vaginal or urethral discharge. So here fixed smears are stained with the Leishman stain and Giardia stain. But here the question is rapid and accurate method. So what is the rapid and accurate method? That is a DFA method. So here that is right answer is option B, DFA, direct fluorescent antibody technique. It is more sensitive method. Next question, infective stage of Trichomonas vaginalis. Option A, trophozoite. Option B, cyst. Option C, precyst. Option D, none of the above. Here the right answer is option A, trophozoite. So trophozoite is the infective form of Trichomonas vaginalis. If you see trophozoite, it is a pear shape. It is having the size of 10 to 30 micrometer in the length and 5 to 20 micrometer in the breadth. So it is motile. Trophozoite is motile by the jerky movements. Next question, parasite causing Dum-dum fever or Asian fever. So which parasite causes Dum-dum fever or Asian parasite? Option A, Leishmania donovani. Option B, Entamoeba histolytica. Option C, Trypanosoma cruzi. Option D, Trypanosoma brucei. Here the right answer is option A, Leishmania donovani. So if you see Leishmania donovani, it was identified by the scientist Sir William Leishman. Sir William Leishman in the year 1900. So he discovered this parasite in the spleen smear of the patient who died of Dum-dum fever at Dum-dum, Kolkata. So this was first identified in the Dum-dum in Kolkata. So it's known as Dum-dum fever. And the same parasite, it was isolated by Donovan, scientist Donovan, in the year 1903, from the spleen smear of the patients from the Chennai. So he did his findings from the London. So this name Leishmania donovani, it is assigned after these two discoverers. So here the right answer is Leishmania donovani. Next question, in stained preparation, Giardia lamblia has dash characteristic appearance. Option A, face-like. Option B, tree-like. Option C, spherical shape. Option D, flower-like. So if you see in the picture, you can see the structure of the Giardia lamblia. So here, what is the right answer? Option A, face-like. So here, Giardia lamblia, it appears like a face-like structure, or like a tennis or badminton racket. So it resembles like tennis or badminton racket, or like face-like structure. Next question, infective stage of Giardia lamblia. Option A, cyst. Option B, trophozoite. Option C, both A and B. That means a cyst and a trophozoite. Option D, none of the above. Here the right answer is option A, cyst. So here the infective stage of Giardia is cyst. So how the humans get the infection? By ingestion of mature cyst in contaminated food and water. The infection occurs by ingestion of mature cyst in contaminated food and water. And what is the infective dose? It is 10 to 100 cysts. Next question. Giardia lamblia inhabits mainly in option A, large intestine, option B, small intestine, option C, respiratory tract, option D, genitourinary tract. So Giardia lamblia is normal inhabitant of option B, small intestine. So it is normally present in the small intestines of humans. Next question. Immunofluorescence assay is done in the stool antigen for detection of Giardia lamblia is by option A, anti-Giardia fluorescence labeled monoclonal antibodies, option B, Giardia antigens, option C, antibody-based antigen, option D, none of the above. So immunofluorescence test is done in the stool by detection of which antibodies? So here the right answer is option A, anti-Giardia fluorescence labeled monoclonal antibodies. Next question. Recently developed antigen test for Giardia lamblia. Option A, Catteridge test. Option B, EIA. Option C, RIA. Option D, none of the above. Here, the right answer is option A, Catteridge test. So Catteridge test is the recent test. Next question. Amoebiasis is transmitted by option A, direct contact with dirty hands. Option B, sexual contact. Option C, fecal contamination of drinking water and food. Option D, all of the above. So here amoebiasis, it is caused by Entamoeba histolytica. So how it is transmitted? So what is the right answer? Option D, all of the above. So it is transmitted by direct contact with the dirty hands, and it is also transmitted by the sexual contact. So how it is transmitted by sexual contact? It is transmitted by orogenital contact or anogenital contact. Especially we can see in the homosexual partners. Option C, fecal contamination of drinking water and food. If the food or water gets contaminated with fecal matter, so if it is consumed by healthy individuals, it leads to the infections. So all these options are true. So what is the right answer? That is option D, all of the above. Next question. Entamoeba histolytica can be cultured in option A, Meconic agar, option B, Cled medium, CLED medium, option C, Diamonds medium, option D, triple N medium, N medium. So here what is the right answer? That is option C, Diamonds medium. So Entamoeba histolytica, it can be cultured in the Diamonds medium. So Diamond in the year 1961, he first reported the culture of Entamoeba histolytica in the absence of bacteria. So in the absence of bacteria, that is called as axenic culture. So axenic culture, it is useful for study of pathogenicity and in vitro anti-amoebic drug testing. So it is useful for pathogenicity testing and in vitro anti-amoebic drug testing and for preparation of axigenic amoebic antigens for the use of immunodiagnosis of amoebiasis. And if you see other options, trade medium, that is CLED medium. What is CLED medium? That is cysteine, lactose, electrolyte deficient agar. So it is for isolation and differentiation of urinary microbes, which causes like urinary tract infections. And if you see option A, Meconic agar, so that is also the for growing of the bacteria, especially for gram-negative bacteria. And if you see triple N medium, that is Navi, McNeil, and Nicole medium, it is for cultivation of Leishmania and trypanosomes. Next question, investigation choice for amoebiasis. So what is the investigation choice for amoebiasis? Option A, ELISA. Option B, microscopy. Option C, RIA, radioimmunoassay. Option D, none of the above. Here, what is the right answer? Option A, ELISA. So ELISA is the diagnostic choice for the amoebiasis. So that is the preferred method. Next question, amoebic dysentery, it differs from bacillary dysentery in all except. So amoebic dysentery is caused by Entamoeba histolytica, and bacillary dysentery is caused by Shigella dysentery. So here, how amoebic dysentery is differ from bacillary dysentery. Option A being offensive, option B dark red, option C acidic, and option D numerous pus cells. Here the right answer is option D, numerous pus cells. So numerous pus cells are present in bacillary dysentery, and pus cells are scanty in case of amoebic dysentery. Next question, infective form of Entamoeba histolytica. What is the infective form of Entamoeba histolytica? Option A, immature uninuclear cyst. Option B, immature binuclear cyst. Option C, mature quadrinucleate cyst. Option D, immature quadrinucleate cyst. Here the right answer is option C, mature quadrinucleate cyst. So mature quadrinucleate cyst, it is the infective cyst which are passed out in the feces of convalescents and carriers. So that is the infective form to the humans. So generally, this quadrinucleate cyst, it contaminates the food as well as the water. Next question, in Entamoeba histolytica, the only one host is option A, dog, option B, monkey, option C, cat, option D, humans. Here the right answer is option D, humans. So humans are the only sources of Entamoeba histolytica. There is no animal reservoir. Next question, accumulation of fluid in the cavity of tunica vaginalis of testis is known as option A, cellulitis, option C, funiculitis, option C, hydrocele, option D, epididymitis. Here the right answer is option C, hydrocele. So accumulation of fluid in the cavity of tunica vaginalis of testis, it is known as hydrocele. And if you see other options, cellulitis, what is meant by cellulitis? It is the deep infection of the skin. And funiculitis, it is the inflammation of the spermatic cord. Funiculitis means inflammation of spermatic cord. And if you see other option, epididymitis, it is the inflammation of epididymis. What is epididymis? It is a narrow, tightly coiled tube that is attached to the each of the testicle. Next question, the causative agent of sleeping sickness is option A, Trypanosoma cruzi, option B, Trypanosoma brucei, option C, Toxoplasma gondii, option D, Leishmania tropica. Here the right answer is option B, Trypanosoma brucei. Trypanosoma brucei is the causative agent of sleeping sickness. Next question, all of the following are the models of transmission of Entamoeba histolytica except option A, fecal-oral route, option B, vectors, option C, sexual route, option D, fomites. So here the right answer is option D, fomites. So as we discussed in the previous slides, it is transmitted by fecal contamination of food and water. So fecal-oral route is correct only. Option B, vectors. So generally houseflies will be the vectors. They are mechanical vectors. And option C, sexual route. So especially we can see in the homosexuals by anogenital or orogenital contact. And Entamoeba histolytica, it is not transmitted by fomites. So what are fomites? Fomites are the inanimate objects. They are intermediate in the transmission of infectious agent. So fomites are clothes or utensils or spoons or any washroom doors or handles. So all these are the fomites, inanimate objects. Next question, clinical features of amoebic liver abscess are all except option A, pain and tenderness in right hypochondrium, option B, quotidian fever, option C, jaundice, option C, emaciated health. So what is the clinical feature of amoebic liver abscess? So here we can see except. So here if you see the right answer is jaundice. So jaundice is not present in case of hepatic liver abscess, amoebic liver abscess. Sorry, jaundice is not seen in case of amoebic liver abscess. But if you see, we can see the symptoms such as pain and tenderness in right hypochondrium, and we can see the quotidian fever. What is quotidian fever? That occurs daily intervals. The fever that occurs daily intervals. And jaundice is not seen in case of amoebic liver abscess. And if you see emaciated health, what is emaciated health? That is severe weight loss. So they severely become thin. Next question. Antony van Leeuwenhoek described dash in his own stool. Option A, Entamoeba histolytica. Option B, Giardia intestinalis. Giardia intestinalis. Option C, Entamoeba coli. Option D, Toxoplasma gonadi. So here the right answer is option B, Giardia intestinalis. So Antony van Leeuwenhoek in the year 1681, he first observed this Giardia intestinalis in his own stools. So it is also called as Giardia lamblia. So this parasite, it is named after Professor Giard of Paris. So it was like named after the Professor Giard of Paris and Professor of Professor Lambert of Prague. He gave the detailed description of parasite. So that's why it is named as Giardia lamblia. Thank you.